Hello and welcome to the Musella Foundation Brain Tumor Webinar Series. Tonight's special guest speaker is Dr. Robert Aiken, a leading neuro-oncologist at Atlantic Health in New Jersey. Dr. Aiken serves as co-director of the Gerald J. Glasser Brain Tumor Center, where he specializes in the diagnosis and management of patients with brain and spinal tumors. Dr. Aiken, please take it away.
Well, thank you very much. I'm going to discuss a phase 2 trial that is open at our site and also, I think, at about half a dozen other sites across the country. It is actually a vaccine trial, a dendritic cell immunotherapy, which is employed in combination with the standard of care for patients with newly diagnosed adult glioblastoma. Treating brain cancer and glioblastoma in particular demands more than a conventional strategy due to a mixture of factors, including the cancer's genetic complexity, invasive growth, immune resistance. New therapeutic approaches involve engineered dendritic cells that mimic the immune response to viral infections. Glioblastoma combines at least 4 distinct challenges that collectively make it one of the most difficult cancers to treat. First, invasion of glioblastoma tumor cells throughout the surrounding brain tissue means that complete resections are generally impossible. Second, significant genetic diversity creates moving targets that can lead to resistance, which often results in failure of traditional approaches in order to achieve long-term treatment success. Third, the blood-brain barrier often limits drug penetration. And finally, the tumor microenvironment is dominated by highly immune suppressive factors, including myeloid cells, that blunt the T cell immunoreactivity, in addition to corticosteroids used to treat cerebral edema, which further dampen the immune response. Dendritic cells are the body's professional antigen presenting cells and are considered the master regulators of adaptive immunity. They detect threats, they process antigens, and ultimately activate a complete adaptive immune response against a perceived threat. They also control the tone, strength, character, and coloration of any downstream response. Research has taught us that properly educated dendritic cells provide the ideal immunotherapy modality for complete and durable immune responses against tumors. Unfortunately, prior attempts with dendritic cells have yielded underwhelming results. alternative immunotherapies have not really had much success in the management of these highly malignant brain tumors. Checkpoint inhibitors like Keytruda and Opdivo, which you've probably been seen advertised on television for a variety of other cancers, these release the brakes on the immune responsiveness, but they only work when there has already been a strong anti-tumor response that has become quiescent, which is not the case in glioblastoma. CAR T cells can be powerful in treating blood cancers, but solid tumors such as glioblastoma present physical barriers, immunosuppressive and acidic environments, and appear to require the targeting of significantly larger number of antigens than is currently possible with the existing CAR T approaches. Peptide or neoantigen vaccines, that is, vaccines that are directed against cancer-related proteins that are expressed on the surface of tumor cells, can be targeted, and they can result in sometimes targeting incorrect antigens. Peptide and messenger RNA tumor vaccines are also not administered in a way that would foster T cell reactivity within the brain tumor tissue. So in summary, the full potential of dendritic cell vaccines has not really reached its maturity to date. The first generation of dendritic cell vaccines consisted of loading tumor lysate, that is, cells that were that were destroyed, and thus the internal antigens could be recognized and picked up by dendritic cells. This, unfortunately, has not really been able to initiate the robust immune response that had been hoped. In addition, messenger RNA loading alone has also not produced the desired benefit. However, the combination of lysate and messenger RNA loading of various tumor antigens, so-called double loading, is the unique aspect of this particular vaccine trial that appears at least promising enough that it has survived a phase 1 trial and is now in a phase 2 trial. This vaccine strategy, this double loading, has been applied to a variety of other tumors, both experimental as well as human. In particular, certain dog species are prone to spontaneously develop brain tumors and hemangiosarcomas. And in those particular de novo naturally occurring tumors, this strategy has resulted in mean overall survival improvement. Glioblastoma has become the major target of my interest, but also there is a clinical trial which is evolving for the treatment of the garden variety and unfortunately still highly malignant and fatal general form of pancreatic cancer. The treatment protocol is relatively straightforward. So surgery is performed in order to harvest tumor tissue. This is sent to the sponsor as fresh frozen tumor tissue. At some point after the surgery and before standard treatment starts, patients have leukapheresis, which is basically designed to obtain peripheral blood monocytes, which are used to generate dendritic cells. Subsequently, and in the laboratory, the tumor tissue is processed with the patient's peripheral blood monocytes, incubated together, and also with the lysate and messenger RNA. These dendritic cells are then what are called double loaded. That is, they are designed to respond to a wide variety of tumor antigens. And then the dendritic cell vaccinations are developed, and patients receive those on a periodic basis prior to the initiation of standard care, which is chemotherapy with temozolomide and concurrent radiotherapy. And then subsequently, standard of care would consist of 6 monthly cycles of temozolomide after the completion of chemotherapy and radiation. So this is a rough schematic of how this process goes. There's the surgical resection and tumor preparation. And then between that and the double loading, patients need to have leukapheresis in order to collect their peripheral blood monocytes. And then that is also dispatched to the sponsor. And then the tumor cells are incubated with the peripheral blood monocytes and also with messenger RNA, which is designed to accentuate the response to certain antigens present on glioblastoma tumor cells. Then this is then vialed and shipped back to us, and then injected every 2 weeks for 3 particular injections. And we know that this induces a robust T cell helper activation via signaling pathways. The injections are actually delivered into the deep cervical lymph nodes, that is, in the neck under ultrasound guidance. So this is not a vaccination that is delivered into a person's arm or leg, but right into the cervical lymph node chain at the base of a person's neck. The phase 1 trial was conducted at both University of Texas in Houston and also an MD Anderson affiliate in Camden, New Jersey. Now, phase 1 trials are usually safety, toxicity, and dose escalation in order to understand what the maximum tolerated dose, or if there is any dose limiting toxicity, is designed to be. And the dose level really, in this case, was the number of cells that could be cultured and subsequently inoculated back into patients. So you can see at dose level 1 and 2, we're talking about 4 patients, and at dose level 3 and 4, there were 10 patients. There was no dose-limiting toxicity. And you can see on the right the schema that was followed. Day 0 was the surgery, leukapheresis, and obtaining monocytes, peripheral blood monocytes, was done in approximately the second week. Subsequently, patients received radiation temozolomide because this period of time allowed the development of these dendritic cell vaccinations, because it does take a period of time in order to generate this particular treatment paradigm. The first inoculation was around day 64, which is about 9 weeks after the initial surgery, with interferon. Interferon is generally designed to stimulate the patient's native immune response. Then the second vaccination is done 2 weeks later, and the third 2 weeks after that. And then patients then enter the monthly cycles of standard temozolomide, which is delivered 5 consecutive days every 4 weeks for 6 months, assuming that they have a favorable response on clinical evaluation and also on periodic imaging. Now, these are this is a swimmer's plot, and broken down by dose level 1, 2, 3, and 4. The 12-month survival versus standard of care was almost 90% versus 60% for standard of care alone. Median overall survival for this very unfavorable group, because the vast majority of these had unmethylated MGMT, was 18.5 months, which is actually, somewhat better than we would have expected, but it certainly wasn't a home run. And here are the imaging responses of 3 people and their tumors. This was the preoperative and postoperative study. And then at 3 months, you can see some increase in the enhancement, 4 months, 5 months, and then at 12 months and subsequently out to 23 months, there is basically no measurable tumor. So this is what we call apparent pseudo-progression. That is, it appears that the tumor is getting worse, but it actually is a response to treatment. That treatment could be the vaccination and or the radiation. So it may be a combination of both. Similarly, in this patient, number 14, original resection, postoperative day 1, and then the evolution of some kind of apparent growth, and then recession of the growth, so that at 18 months, there is no apparent evidence of tumor. And similarly, a recurrent patient who had an unmethylated MGMT, again, a very unfavorable patient group. Similarly, apparent progression and then remission. This is a nice pictorial which demonstrates the presence of the increase in CD8 T cells after treatment, which is clearly not present before treatment. And these are the immunologically active tumor cells, T cells, which are probably doing the heavy lifting in terms of treatment. And these can be quantified both pre-vaccination and post-vaccination, so that you can actually see the median rises to some degree in both CD8 and CD4 T cells. Now, this is a overall survival plot. And the important information here, this is basically a statistical overall survival with standard of care. But the important thing that I want to show you on this particular graph is that patients who had subsequent surgery to evaluate the result of this apparent treatment response or tumor growth actually did less well than patients who were just observed after surgery and did not have any subsequent biopsy in order to identify if this was true tumor or treatment effect. The reason for this is purely speculative at this point in time, but it may have to do with the fact that patients who had surgery, more likely than not, were also receiving corticosteroids, which have an immune blunting effect, as opposed to those people who received the vaccine only and did not have rebiopsy or re-resection in those situations where it appeared that there might be tumor progression. Side effects. Again, this is all from the small number of people in the phase 1 clinical trial. Most of the side effects were really mild and quite self-limited, and those that you would probably expect from a vaccine: fatigue, chills, and some degree of a rash. Flu-like symptoms, generally quite mild. So this brings us to the phase 2 trial, which we are participating in. So patients with newly diagnosed glioblastoma are then invited to participate in the trial. They need to have tissue obtained at the time of surgery, which is then dispatched to the sponsor, and that tumor tissue is stored and sent on the day of surgery to the sponsor, which is in Houston. Patients at the end of their chemotherapy radiation, the standard 6 weeks of chemotherapy radiation, are then randomized either to arm A, which is the active vaccine plus standard of care, or to arm B, which is standard of care only. This is a 1-to-1 randomization for the first 60 patients. The endpoints are the typical endpoints that we have adopted for the management of patients with this tumor. The primary endpoint being overall survival, secondary endpoints being safety, progression-free survival, time to tumor progression, and then there are exploratory endpoints where we are looking at health-related quality of life, neurocognitive function, immune biomarkers, and noting the rate and frequency of pseudo-progression. Again, the rough schematic to your right: surgery, about week 4 is leukapheresis. Weeks 5 through 10 are the radiochemotherapy with temozolomide. And then following radiochemotherapy, patients will receive the injections into the cervical lymph node chain with PEG interferon every 2 weeks. And then they are followed for evidence of response or failure. The first 60 patients are generally going to be randomized 1-to-1. After the first 60 patients, and assuming safety, tolerability, and potentially some benefit on the initial 60 patient look back, if the trial is allowed to go forward, there will be a 2-to-1 randomization for the remaining 90 patients or 150 patients in the clinical trial altogether. Now, there will be what we call a Bayesian adjustment to randomization, which means that there is a continuous look back and that patients will be assigned to the treatment arm, perhaps more based on the prior responsiveness of individuals who have participated in the phase 2 and in the phase 3 trial, which would arise after 150 patients and assuming that there is evidence of efficacy at that point. Inclusion criteria: adults 18 and older, a willingness to comply with all of the study procedures, including willingness to understand and participate if they are randomized to the standard of care. Patients really need to have a gross total resection or a near gross total resection, which is defined as at least 70% of the contrast enhancing tumor. They can be randomized on the basis of IDH wild type, which is detected usually after surgery by an immunohistochemical assay, but subsequently is revised based on the next generation tumor sequencing. And if IDH mutation is present, then they are excluded from the trial. So these are only patients who have wild type IDH expression in their tumor. Extent of resection and non-enhancing disease, I think I'll skip over that. And again, molecular testing is usually something that does not get reported back except during patients' chemoradiation. And so there is the opportunity to revise their participation in the trial if they possess an IDH mutation on the next generation tumor sequencing. Again, they also need to receive Neupogen as part of the leukapheresis process, and then the biweekly injections of the vaccine, the dendritic cell vaccine, followed by the interferon inoculations as well. Women of reproductive potential must have repeatedly negative serum pregnancy tests and agree to use at least 2 forms of contraception, minimum liver, renal, and bone marrow function, as well as immunological function, and have a certain level of performance at the time of surgery. Exclusion criteria are fairly standard. Leptomeningeal disease, tumors that have spread outside of the brain, which are exceedingly rare in the case of glioblastoma, patients who have a history of HIV or who have Hep V, Hep C, I'm sorry, or autoimmune disorders are excluded from the trial. I'll take any questions.
Thank you for that presentation, Dr. Aiken. We have quite a few questions, so I'll try and round them up. So you did mention that it was one-to-one randomization for the first 60 enrolled, but I think that the trial's been active for quite a few months now. Do you know what the enrollment status is as of now?
It's still 1-to-1, but I think that they are getting close to that 60 mark.
Okay. And for the limit on residual tumor that you mentioned, why is it such a strict limit? And do you think the vaccine would still provide a survival benefit even if a patient had significant residual tumor? Just
Well,
theoretically.
that's always the $64 question. And generally speaking, when we're talking about immune treatments, we're talking about maximal cytoreductive surgeries because we recognize that despite the um, progress that we appear to be making in understanding the immunobiology of cancer, the treatments are still really much more effective in patients who have had a maximum cytoreductive surgery at this particular point. These tumors are immunologically very, very cold. And so it really is to a patient's advantage to have a maximum or gross total resection, or failing that, having a at least greater than 70% resection of their tumor. At some point in the future, when our therapies may be more effective, we may be able to talk about treatment after, let's say, biopsy only, or even treatment based on inferred diagnosis. But I think right now, these are really patients who need to have a very skilled neurosurgical resection.
Okay. Does the trial allow Optune? And also, do you think there could be synergy between Optune and this vaccine?
At the present time, the trial does not allow for Optune. Um, the reason for that is that in phase 2 trials at this point, and the way they are designed and the way they are approved by the FDA, is that there has to be unequivocal evidence of response to the treatment under investigation. Um, Clearly, if something like this were to be approved, there would be no doubt the desire to do both. And that is an uncontrolled environment, so that there would certainly be patients who would probably choose to do both, figuring that there may be synergy, but again, this is not known, and it could very well be subject of an additional trial uh, after it is, if it is approved, let's say.
Yeah, of course, the patient advocacy standpoint, we always like to see Optune allowed. And, in an ideal world, you want to compare your investigational treatment to the best of the available treatments, which would include Optune, but definitely understand the constraints. You
Right.
mentioned,
These are really constraints that are imposed by the FDA in order to prove with beyond a statistical doubt that the treatment under question is effective.
mhm. And you mentioned in the trial there are 3 vaccine doses given. Why not more?
Um, that's a very good question. And I think it really has to do with vaccine availability, because we're talking about harvesting a certain number of cells. And so the limit is really imposed on the basis of the number of cells, the dendritic cell numbers that can be generated, um, so that at this point, these tumor cells are not grown in cell culture to expand the number of cells that are harvested. Um, so there is really a limit based on the number of tumor cells that are obtained and then can be cultured and dendritic cell um, vaccine generated on the basis of a limited number of cells.
Okay. Could you potentially do leukapheresis more than once to generate more?
That's not the rate limiting factor. The rate limiting factor is the number of cells that are
No.
generated by this tumor resection.
Okay. You had that great slide with the three patients and kind of the pseudo-progression over
Yes.
time. I'm really curious for the pseudo-progression seen in this in the phase one and now in the phase two. Does it always sort of resolve by the 12-month mark, or what is the time frame during which you're seeing pseudo-progression?
Well, usually pseudoprogression from radiation reaches its maximum between 6 and 8 months. The additional variable here is the immune responsiveness of the vaccination schema. So it is really unclear at this particular point whether or not this is something that is just due to the radiation, or is radiation plus vaccination. Clearly, neuro-oncologists see pseudo-progression all of the time. And so it isn't clear, based on three examples, whether or not this is a common occurrence or an uncommon occurrence.
Okay, and for the pseudo-progression, because you mentioned that some patients had re-resection in the phase one and they didn't do as well as those who didn't have
That's
a re-resection. So
yeah,
have
that
you found other ways of managing potential pseudo-progression? For example, obviously dexamethasone is immunosuppressive. So have you
right.
been using Avastin as a temporary band-aid to control that swelling?
Well, I haven't seen that yet. So the question in this situation is, it wasn't clear what the management of those patients was. It did not appear, on the basis of the three examples that I presented, that the apparent pseudo progression versus progression was very severe. So it is quite conceivable that because these patients were in a clinical trial, and because the clinical trial probably made was with skilled neuro-oncologists who routinely know how to interpret both patient responses as well as MRI uh, evidence, that these patients could very well have been followed rather frequently during this period of uncertainty, until it was evident that this process was resolving. Again, these are studies that are done in brain tumor centers, where there is a lot of experience in managing patients through periods of uncertainty.
Absolutely. Do you think this vaccine would work in recurrent GBM, and will there be a trial for recurrent GBM patients in the future?
I'm sure that if this uh, tumor strategy is approved for newly diagnosed, it's only a matter of time before it is explored in the setting of recurrent disease.
And currently is for patients who are recurrent or patients who don't otherwise meet the inclusion-exclusion criteria, is there any way for them to access this vaccine platform outside of trial?
No.
Okay. And I've had a couple different people ask about a comparison between this vaccine platform and the tumor lysate vaccine DC-Vax-L.
Right. Well, remember that first slide that I showed, which was the tumor lysate only. That was the DC-Vax strategy, was tumor lysate only, co-incubated with dendritic cells that were harvested through leukapheresis. And unfortunately, the DC-Vax story does not appear to be ending well at this particular point in time. It has not been approved in any country. And I think that it is clearly a first generation vaccine. And now we're probably in third and fourth generation vaccines. So it appears, at least based on my reading of the evidence to date, that DC-Vax is unlikely to see the light of day in this country. I can't swear for perhaps other countries.
And I'll just speak from the patient advocacy point of view. We do hope to see its approval in the UK and other places just because it's had a successful phase 3 and that's so rare for GBM.
Right.
Uh,
There were issues with regards to crossover, responsiveness, but I agree. I think that there is compelling evidence of some benefit, and it is actually being used off-label in the UK.
yeah, and I do think its approval would help pave the way for these second and third generation vaccine platforms. Okay, you mentioned you give the interferon injections as well. How did you choose that as kind of your immune booster, and did you consider other ones?
Well, let me say first that I didn't choose it. This is part of the treatment protocol, but using gamma interferon, PEG interferon, is a standard way to basically augment a immunostimulatory effect. So it is actually a reasonably effective way of augmenting an otherwise modest immune response.
And along those lines, do you think there will be plans to explore the vaccine together with checkpoint inhibitors in the future?
I think that once the strategy reaches a point where it appears that it is more likely to be effective than not, and that is always, a moving target based on the phase 2 and phase 3 portions of the clinical trial. If there is evidence that that appears to be promising, I have no doubt that there will be ways invented or conceived of that will further enhance an immunostimulatory model.
Okay. I don't know if you're familiar with IOZK in Germany, but someone asked, how does this compare to the approach taken by IOZK in Germany?
I'm not familiar with that,
Okay.
unless are you talking
Well,
about
well,
CGAT?
no, it's actually a different clinic, different multimodal strategy. So we can skip that one.
Okay.
Um, okay. Can this vaccine be used on other brain tumors besides GBM? And I'm going to wrap this in with another question specifically about IDH mutant tumors. Would this work for IDH mutant tumors? Would this work for other kinds of brain tumors?
Well, there's no reason why it couldn't. It's just that at this point in time, this is the restriction imposed by the trial, that we are only looking at IDH wild type, um, newly diagnosed glioblastoma.
Okay. I think that's all of the questions, unless Al, if you're on and you're listening, if you had any more, I think that was all the questions I got.
No, that was it. Very interesting presentation.
Okay. Well, it
Thank
was
you.
nice to see you all.
It's very impressive, those long-term results, especially in the unmethylated patients,
Yes. And this is
because another option for unmethylated.
that's right. Unmethylated patients generally have a much worse trajectory.
Okay, thank you, and good night, everybody.
Thank you.
See you next time.
Good night.
Thank you.